Okay, so excited to have today's talk featuring a topic that I'm really interested in learning more about and promises to be fascinating. Genomic epidemiology on the front line and learning about disease transmission from pathogen genome sequences by Dr. Allison Black from the Bedford Lab at Fred Hutch. And let's see. Just a quick reminder to mute yourself if you're not talking. Feel free to add your questions into the chat. And we do have a feedback form at the end of the talk that really helps us think about this format and how we can serve you better during this time. And it's my great pleasure to introduce Dr. Allison Black. She is at the Department of Epidemiology at the University of Washington, and also, as I mentioned, in Trevor Bedford's lab at Fred Hutch. And I am going to stop sharing my screen so that she can introduce herself and start sharing her screen.
Awesome, thank you. All right, I will do this. Can everyone see my title slide? Perfect.
Yes.
Okay, fabulous. Well, thank you all so much for having me. It's a real pleasure to talk with you guys, especially because I had excellent teachers basically all the way through. And so I'm excited to get to share the science that we've been working on with all of you. I was asked to kind of start off by just talking about my path to where I am now. And interestingly, this actually begins with TV for me. So if this looks to you like late 90s, early 2000s television, that's because it is. There was this show called Medical Detectives on the Learning Channel. And I love this show. I actually am Canadian. I grew up in a rural part of British Columbia, and we didn't have cable. And so when I would go to my grandmother's house, she did, and it was like a big treat to watch TV. And I would get to watch this show that I absolutely loved. And it was a lot of sort of medical mysteries about the clinical courses of disease, but also tapping into diseases at a population level as well. And one of the episodes I was particularly interested in investigated the transmission of bovine spongiform encephalopathy, BSE, or what's better known as mad cow disease in the UK, and really looking at the emergence of the syndrome as well as the transmission of the prion. And I thought this was just so cool. It was this mix of science and medicine with really just hardcore detective work and sleuthing. And I talked a lot about it with my mom, who was a hospital pharmacist. And she was like, well, maybe you should like look into epidemiology. And I was like, oh yeah, maybe. As you know, a sort of high schooler does, and then goes on and looks at a whole bunch of other careers. But in fact, the seed was planted. And I have always been interested in disease, and I still am. And lo and behold, I have become an epidemiologist. So in high school, I studied a lot of chemistry and physics and math, and I actually stopped taking biology after grade 10. And I think something that's really interesting is if you just build, or at least what I've found is if you build a wide enough base in science generally, you can return to bits of science that you've left behind. And so this is something that I started doing in undergrad. I mentioned that I'm Canadian. I went to a liberal arts and sciences university up in Squamish, BC, which is just sort of 40 minutes up the road from Vancouver. And this was great. I did a very interdisciplinary degree. I studied physics and math and political science and philosophy. And this was when I really got back into biology. I was still really interested in disease. And primarily the social factors that facilitate disease transmission. But I ended up working with this amazing professor, Annie Prudhomme-Genereux, down here. And she was a biologist, and she got me really interested in evolution and biochem and all of these sort of biology-oriented questions about pathogens. And that really led me to consider doing my master's degree in microbiology. And I went out to Winnipeg, which is where the National Microbiology Laboratories in Canada are. And it's also probably like the coldest place you can live in Canada, which is I think why they put this hardcore lab out there where they study all of the select agent pathogens. So kind of fun fact, this is where the Ebola virus vaccine was actually developed at the National Microbiology Laboratories in Canada. And I put this in here because this was a really great learning experience for me, but it also was not your sort of stereotypical path. So I went out to Manitoba thinking that I would do my master's in microbiology. And I started out doing that and realized that as much as I loved learning about viral genetics and sort of molecular biology of pathogens, I really did not want to have the day-to-day life of a microbiologist. I was still really interested in the population level dynamics of diseases and less interested in figuring out the actual sort of mechanisms of replication. And that was really what sort of lab science microbiology was oriented towards. So I ended up leaving. I did not pursue my master's here. And I ended up back in Vancouver, near to where I'm from, working at the BC Centre for Disease Control. And it was here that I was mentored by Jennifer Gardy, who's actually now at the Gates Foundation. But at the time, she was an epidemiologist working on tuberculosis here up in Vancouver. And she worked in this really cool realm of epidemiology that combined my two passions in terms of understanding pathogen genetics and pathogen molecular biology with epidemiology and understanding diseases at the population level. And she kind of helped me to realize that there was this entire world of what we call genomic epidemiology. And that's really what got me started on the path that I'm on now. I ended up coming to the University of Washington to do my master's in genetic epidemiology at the University of Washington. And then I met my current advisor, Trevor Bedford, and I stayed on to do my PhD. And sort of the rest is history. And it's one of these things where you can connect the dots looking backwards, but it felt like a topsy-turvy path where I was sort of ricocheting between things and trying to figure out what worked and what didn't as I went through it. So transitioning to the work I've been doing, I wanted to kind of open things off with this image, which some of you might recognize, because this is sort of an encapsulating image of what epidemiology is. So this is John Snow's map of cholera fatalities in London. John Snow is considered the father of modern epidemiology, and really it was because that he did this mechanistic investigation into disease transmission. At the time, most people thought that cholera was spread through miasma, but he had a hypothesis that it was in fact waterborne. And to investigate this, he was interested in cholera fatalities resulting from contaminated water sourced from the Broad Street pump. So each of these black bars represents a cholera fatality, and they're stacked when they occur in the same household. So you can see here this household of many fatalities. And what he noticed by mapping the spatial distribution of cholera fatalities was that they tended to cluster around the Broad Street pump. So this is a very typical question that we would ask in a descriptive epidemiology study, which is the typical type of study that we do when we have a new outbreak and we're wanting to understand it, where we say, or where we ask questions about who is getting infected and where they're getting infected, and when, and all of these sort of descriptive questions of what's going on. Those initial descriptive studies tend to prompt hypotheses that then we test and explore with analytical epidemiology studies. And this is sort of what John Snow did. He hypothesized that it was related to water sourced from the Broad Street pump. And he went and he removed the handle off of the pump so that people couldn't get water from there. And he monitored the fatalities of cholera after that intervention. And that is really what we're trying to do in epidemiology. We're trying to understand disease such that we can design effective interventions to limit it. This is not exactly how we do data all the time now in epidemiology. So what is a more traditional picture is actually to, instead of looking at case counts through space, we generally, in modern infectious disease epidemiology, look at case counts through time. So this here in the middle is what we call an epidemiologic curve, and it plots the numbers of cases over time. So you can see you have one back here, and then kind of the epidemic grows, and then it descends. We can tell a lot about the dynamics of a disease from the shapes of these curves. So if you have propagated transmission from people kind of going onward, that will yield a different shape compared to something where, so you say, have a point source exposure where everyone goes to a picnic and they eat some bad potato salad and they all end up sick at the same time, but there's not really any onward transmission. So those shapes are important. You can do things like estimate the rate of epidemic growth from these curves. And that is really the heart of modern surveillance epidemiology, is looking at case counts of disease. And genomic epidemiology kind of looks at this even a step deeper, and it says, okay, for each of these people that get recorded in this epidemiologic curve, you have a person that became infected and who possibly passed along the infection to someone else. And so instead of looking just at the people and counting them, let's look at the pathogens themselves that have been transmitted between these people, because probably there will be some sort of recording of the transmission process within that pathogen itself. You can do this with bacterial or parasitic or viral pathogens. I'm going to talk about viruses because that is what I have studied during all of my dissertation work. Okay, so how does this work? How do we look at transmission histories based off of the pathogens instead of the people? Similarly, I think probably many of you have played telephone with kids before, and you sit them in a line. You have one kid start, give them a word, and they pass it along, and the word sort of morphs as it is heard differently by different kids. And viruses actually do something very similar. So when you are infected with a virus, that virus enters into your cells and it starts to try to make progeny viruses. It replicates. Most viruses, though, are kind of sloppy. They make mistakes, and they don't have ways to fix those mistakes as they're copying their genome sequences. And so what that means is, over the course of an infection, a single person will have all of these progeny viruses within their bodies that will have slightly different genome sequences. A lot of these changes will make the virus non-viable. It won't, it'll die. It won't be able to infect cells. Some of them will have absolutely no functional change whatsoever. Some of them might be beneficial, but by and large, it's just sort of a random thing like a typo. And every now and then, a virus that is ever so slightly different, that has this new variation, will rise in frequency so that we see it. And what that means then is that over time, as person 1 transmits their virus to person 2, at some point, one of the changes that accrues over the course of the infection rises up in frequency. And we can see that person 3 has a slightly different sequence variant that they then transmit on to person 4. When you take viruses from all of these different people and you sequence the viruses and you look at those genome sequences, you make what we call an alignment, you can see how all of those different nucleotides or those variants map across a big scale, where you have perhaps hundreds of samples, and usually these sequences are thousands of nucleotides long. And you can do that and you can say, okay, well, given the sequence similarity, I think that these two cases are most closely related. So we go from building this process of a transmission chain to having a structure that we call a phylogeny. This is basically just a family tree of viruses. So these two individuals, when we sample them, they have the same genome sequence. They both share a G at this last site. And so they are more closely related to each other than these two viruses over here, the blue ones, that have an A at that last site. We can kind of map back what the history of this mutation process looks like, and we can say, okay, well, given that there was an ancestral virus sometime back here that likely infected these people, what sequence do I think they would have? And given that both of the descendants have a G at that site, I think that this unknown, unsampled ancestor also likely had a G. And you can reconstruct that history probabilistically over the phylogeny. And then you can say, okay, well, I think that this event where I had a mutation from G to A occurred along this branch here. And that the ancestor back here had a G at that site at some probability, and that turned to an A, and that's how I ended up with these cases that are most closely related. So this reconstruction of the sequence is useful, but it's a lot more useful when we combine it with other epidemiologic data. And this is really what we're doing, where we don't just have the viruses from people, but we have characteristics about the samples that were collected as well. So we often know where people lived and also the date that their sample was taken. And so similarly to how we reconstructed the sequences of ancestral viruses, we can ask sort of similar questions about geography, where both of these people who were infected with red viruses live in Canada. And so we can say, well, given that both of those people with the same viruses were living in Canada, probably this ancestor also circulated in Canada. And probably this ancestor circulated in Canada too, with some sort of quantifiable probability. And yet these blue viruses infected individuals who were living both in Switzerland. And thus at some point, the virus must have jumped from Canada to Switzerland. And given that the ancestor here was probably in Canada and the ancestor here is probably in Switzerland, that event probably happened some point along this branch of the tree. That branch of the tree can actually be translated into a time. So when we have the genome sequences as well as the dates that a sample is collected, we can start to estimate how quickly pathogens are evolving, or what we call the evolutionary rate. And thus, given the amount of sequence distance that there are between two viruses, we can actually guess as to how much time separates them. And you get what we call these temporally resolved phylogenies, where the branch lengths actually represent calendar time. And so not only do we know that we had a likely event where the virus moved from Canada to Switzerland, we can actually start to date when that happened. And so this is just a toy example of what this process looks like. But we do this when we have hundreds of sequences and many sites and many countries. And it's with this kind of process that we start to map out the descriptive epidemiology of pathogens in terms of who do they infect, which countries are they moving between, which countries tend to share outbreaks, are there countries that tend to be the source of disease more frequently, and are there countries that tend to be the sink, and when are these introductions happening, and how did they grow and expand? And that is really the heart of genomic epidemiology. And I'm going to be talking about those processes and the findings that we have inferred from these types of analyses, from sequence data from Zika virus, from Ebola virus, and then I'm going to talk briefly at the end about SARS-CoV-2, which is the virus causing COVID-19. So I'm going to start off talking about Zika virus. I've studied Zika for most of my PhD, I would say. I think when you are an epidemiologist working on emerging infectious diseases, basically you look for whatever outbreak is happening when you start, and you spend a lot of time trying to understand that thing. And 5 years ago when I started my PhD, it was Zika virus that had emerged in the Americas, and there were all of these open questions about how long it had circulated, how it came to Brazil, and what had happened after that fact. What a lot of people don't know is that Colombia actually had the highest numbers of cases, or reported cases, after Brazil. But there wasn't as detailed of an investigation as to what occurred in Colombia compared to Brazil. And so we sought to do a genomic epidemiologic study on sequence viruses from Colombia to better understand what the epidemiologic dynamics in that country were like. And this is work that I actually co-led with a postdoc in our lab, Louise and I. So our central questions then were, how many times was Zika virus introduced into Colombia? So you can imagine that you could On one extreme, have a single introduction that yields all of this downstream transmission within the country. Or you could have a pattern where you in fact have lots of different introduction events as people travel to other affected countries and come and bring back the virus to Colombia. And disentangling those is important because it might actually change how you are going to address limiting the outbreak, whether you need to focus more on what's happening within your country or whether you need to stop these travel-associated importations. We also wanted to know when introductions were occurring. And this is a particularly important question with Zika virus because so many Zika virus infections are in fact asymptomatic. And so what we noticed in Brazil is that Zika virus circulated in Brazil for a year before we realized that it was even there. And so dating when those things actually arrived is important because it gives us a better understanding of when people are at risk of Zika virus infection, and thus also when we need to be looking for elevated risks of sequelae from Zika virus infection, such as Guillain-Barré syndrome or microcephalic pregnancies, or anything like that. So it's actually a really important quantity for all of these later epidemiologic studies that occurred trying to understand the role of Zika virus infection in microcephaly and things like that. And then, as I alluded to earlier when I was talking about introductions, we have this broader question of, okay, well, what does this timing and frequency tell us about how the epidemic was actually sustained in Colombia? In practice, we went about collecting data by packing up our molecular biology reagents and plastics in a bunch of suitcases and flying them down to Bogotá. We were working with the National Health Institute down there, and we spent 3 weeks in Bogotá looking for samples in this kind of hall of freezers and doing a whole lot of lab work to extract viruses from clinical diagnostic specimens that had been collected for the purpose of determining whether people had Zika or not, and trying to get virus out of them and sequence it. So Zika is a particularly challenging virus to sequence. And in the end, despite all of our best efforts, we were actually only able to obtain 8 sequences. But luckily, these came from a very diverse area across Colombia. So prior to our study, there had been other sequences collected, but they were primarily from Barranquilla, which is kind of up here on the Caribbean coast. And so we were able to sample a greater swath of Colombia. Once we had those sequences, we combined them with the other publicly available sequences from Colombia, and then sequences that were available from many other countries that were affected with Zika in the Americas. And we built these large phylogenetic trees. So we can see up here, this is a temporally resolved tree where these branch lengths are telling me something about calendar time, just like in the toy example. And then where the coloring on the branches tells me something about where we infer the ancestral viruses to have circulated. So the tips are sampled, we know where they came from, and then anything on the interior where these branches are colored tells us something about where probabilistically we think that the ancestral viruses circulated. When we look at the Zika virus epidemic with this kind of view, what we see is that there are in fact 2 groups of viruses circulating in Colombia that are quite diverged. So we have this first introduction, which goes from Brazil into Colombia, and that it's actually quite distant from this other clade of viruses that was also introduced from Brazil into Colombia. So zooming in on those 2 different groups, I'm going to start with clade 1, which is the larger clade. So given the rate of evolution and the amount of distance between the genome sequences of all of these different viruses, we inferred that this clade, or this group, was introduced to Colombia back in February of 2015. And what's interesting about this is that this is about 5 to 8 months before there was confirmed circulation of Zika virus in Colombia. And it's actually even earlier than the confirmation of Zika virus circulation in Brazil. So we know that Zika circulated in Brazil for a long time before we detected it formally. And what this indicates is that we actually had transmission out of Brazil into other South American countries before we even knew that it was in the Americas. So you can look at this and you can say that there's actually a fair amount of diversity between the Colombian samples. And so given this process where we know that when you have an infection, you get the sort of diversification of the virus, and then sometimes that mutation is passed along, what we can see is that there's a fair amount of this Colombian outbreak. There's a fair amount of transmission that is captured in this clade. And so really the vast majority of transmission within Colombia appears to descend from this single introduction. Another thing that we can note is that there is a lot of transmission out of Colombia into other countries that generally share a border with Colombia. So you can see here there's an exportation, the virus is circulating in Colombia, and it moves from Colombia into Panama. Seeing here, it's circulating in Colombia and it moves into Peru or into Martinique or into the Dominican Republic. And so what we can see here is that there's transmission, there is an introduction event into Colombia. We see transmission occurring within Colombia, and then the exportation of Zika virus into other neighboring countries that starts their epidemics as well. If we look at this smaller clade, there were only 3 of the 20 Colombian genomes that grouped within this clade. And what that means then is that the vast majority of viruses were related to the other introduction. But this was a new introduction that we hadn't seen before our study. And I think that the reason that we hadn't seen it previously is because so many of the other sequencing efforts had been really focused on 1 or 2 cities. And when we got a broader sampling across the country, we were able to more randomly sample the diversity of viruses that had arrived and circulated in Colombia. Again, we see sort of a similar pattern as before, where we have transmission within Colombia and then movement of the virus into neighboring Ecuador. And this introduction occurred a little bit later in September of 2015. So taken all together, we estimate that there were at least 2 introductions of Zika virus into Colombia, although it's possible that if we sequenced more, we would find a few more introductions. That the primary introduction was introduced into Colombia about 5 to 8 months prior to the confirmation of Zika circulation in Colombia. And that's really important. Colombia was one of the countries where we did a lot of studies looking at Zika virus sequelae, including the risk of Guillain-Barré among individuals who had been infected with Zika. And in order to do those studies in a reliable fashion, if you are comparing the rates of Guillain-Barré, you want to compare it at a time when you don't have the risk factor, which is Zika virus, compared to the incidence of Guillain-Barré when you do have the risk factor, which is Zika virus circulation. And so conducting those studies well requires that you know when Zika was actually present in the country. So the final point is that the Colombian epidemic was primarily sustained by within-country transmission rather than by reseeding from other countries with their own outbreaks. And so really what this means is that if we could have done a better job with vector control, probably of early surveillance and knowing about Zika virus, that the scale of the epidemic in Colombia probably could have been curtailed. I just want to, should I be answering questions in the chat as I go, or?
There aren't any yet, so we're good. I'd like to
message, I think, from Eric. Okay, I'll respond to this really quick because I have this open. Eric asked me, how does this gathering of information help us plan for the future? Basically, it helps us to understand what types of dynamics tend to facilitate vector-borne disease transmission. And you know, we know from chikungunya as well as dengue and now Zika, there's been sort of continual reemergence of vector-borne transmission. And so I think that it gives us an understanding of what those epidemiologic dynamics could be and how to surveil for them better. It also helps us to understand how widespread some of these outbreaks might be. So how many people might have immunity to Zika. And that allows us, when we're doing risk modeling, to try and understand when future outbreaks might occur. Knowing how many people were affected and immune, and knowing sort of those rates at which point there will be enough susceptible people to sustain another outbreak, sort of lets us know when we might start expecting more Zika virus outbreaks and kind of prepare in that way.
So for folks who don't know, could you explain a little bit what vector-based dynamics mean?
Yeah, definitely. So Zika virus is, it spreads because a mosquito bites an infected person or an infected animal, and the mosquito becomes infected and then bites someone else, gives that person the virus, and then another mosquito bites the infected person, the mosquito becomes infected. And so you have this cycle where instead of people directly giving the disease to each other, you have what we call vector, and this might be a tick, or it might be a mosquito, being sort of the intermediate step. So you can't have a person giving Zika, or the vast majority of the time, we don't think that people can directly give each other Zika, but instead it requires this, this intermediate host, the mosquito, to transmit it.
And then there was a question, do you need the entire genome sequence in order to place it on your tree?
Oh, that is such a good question. So no, you don't, but there's nuance to that. So you can place shorter sequences on there. But what happens with these models is that anytime you have a sequence that sort of doesn't have nucleotides at a site in the genome, it treats it as unknown. And so when you have shorter sequences, you just have less, like you have sort of less slots where you're looking for nucleotide mutations. And what that means is that you actually just have less signal to detect them. And so when you are reconstructing those trees, you just have sort of the shorter sequences have less information, and it's harder to place them. You don't really know where they belong because you don't have any information at many sites where you might be looking for SNPs. And so they can sometimes fall in unexpected places on the tree, and that's something you kind of have to watch for in analyses. And so I generally tended to use, for these analyses specifically, sequences that were at least 80% full.
Great, thank you.
You bet. All right, so jumping back to the talk and shifting gears, I'm going to talk a little bit about work that we've been doing, helping to support efforts led by the National Biomedical Research Agency in the Congo, monitoring and understanding the epidemiology of the Ebola outbreak that's going on right now in the Nord-Kivu area, which is in the eastern part of the Congo. So Ebola was actually discovered in the Congo back when it was referred to as Zaire. And the Congo is no stranger to Ebola. This is in fact their 10th outbreak, but it is the largest one. So this current outbreak has been ongoing since 2018. It's caused almost 3,500 cases and over 2,000 deaths. And I think something that's particularly startling about this is that this is in the age of an Ebola vaccine that was tested in West Africa that we know is highly effective. And so, you know, there are these standing questions about what factors are facilitating Ebola transmission. Probably a lot of the issue with the vaccine is just that people are refusing to get it. But if they are refusing to get it, you still need to control Ebola with your traditional measures, which epidemiologically speaking, this is when you use contact tracing. So if you have a person who tests positive for Ebola, you ask them about all of the people who they interacted with, and you ask all of those people or the contacts to monitor themselves for symptoms. And the goal then is that you're trying to isolate people as soon as they develop symptoms so that they can't pass the disease on. And this is very, very manual work. You end up, I think like there was tens of thousands of people under monitoring. And so the kind of public health surveillance aspect of this is very, very intensive. And we've actually been trying to support this not only with case-based surveillance, which is what I'm showing here with these epidemiologic curves, but also with genomic surveillance. So throughout the outbreak, the INRB has been, which is the National Biomedical Research Agency in the Congo, has been conducting genomic surveillance. So in gray, I have plotted out the PCR confirmed cases of Ebola virus disease. over time. And in orange, it shows the fraction of those cases that were actually sequenced. And you can see here that the sequencing has really been occurring throughout the entire outbreak, and I can't stress how unique and special that is. So the INRB has been sequencing roughly 15% of confirmed cases. And just for context, during the West African outbreak, it was considered the most deeply sequenced outbreak we've had to date, and they had 5% of confirmed cases sequenced. So really building capacity, shouldn't be a surprise, but building capacity within the country is really the best way to have these strong, sustainable systems that actually work much better for collecting public health data. So just doing the sequencing though isn't quite enough, and there's a lot of analysis to this as well, and it's tricky and it's data intensive, and that's kind of becoming one of the harder parts of this work. Is not just necessarily generating sequence data, but analyzing it and doing that in a timely manner. And this is where we came in to help support INRB. So prior to September 2019, these curves are showing the amount of time it took between different events. So in orange, we have the amount of time that passed between when a sample was taken from a person and when it was sequenced. In teal, the time between when the sequence was generated and when it was released privately to public health partners. And then in purple, the time between sequencing and public release. And you can see that these lag times are actually really long in some cases, like 10+ weeks, which is not fast enough to be helping guide response efforts in real time. In September 2019, I and a postdoc in our lab, James Hadfield, traveled to Kinshasa to bring computers and software for doing this analysis, and also train Congolese scientists to be able to do the bioinformatic work themselves within Kinshasa. And this has made a huge amount of difference. So now that the data analysis actually happens in country, along with the sequencing, the lag time between sequencing and private data release to frontline public health workers is much, much shorter. The lag time between sample and sequencing is still relatively long. It's hard to bring that down because this is just a really challenging environment to work in with a lot of violence that can shut down labs and affect sampling, and it's tricky. There's lots of things to talk about there that I'm not going to go into detail here for the sake of time. But what this speed up has meant is that we can now analyze genomic results quickly enough that we can get those inferences back to frontline public health workers. And what we find in the data can actually inform how they tailor their response efforts. So these are some examples of what we monitor the Ebola genomic data for. We look for super spreading events. So this one was related to an unsafe burial of a pastor that was widely attended and resulted in a ton of secondary cases directly related to having contact with the deceased, but then just a huge amount of onward transmission that was sustained and sort of reignited the outbreak. We also use genomic data to look at possible instances of sexual transmission. And this can be challenging to pick up when just looking at the case surveillance data, because this case will just show up looking like any other case of Ebola. But when you look at the genome sequence, what we see with sexually transmitted Ebola cases is that the viruses tend to look much older than they are, or they have much fewer mutations that you would expect. And so you get this clear signal that alerts you that something is a little different. And that's important because certainly these sexual transmission cases can kind of reignite outbreaks that had otherwise been relatively well controlled. And we also help monitor for cryptic transmission. So looking for cases or transmission chains that are circulating but undetected. And this is really critical when you're trying to use contact tracing to control a disease. You're only as able to control it as you know about all of the people that have it. And so we can use the genomic data as a record of a transmission history that has happened, even if people don't necessarily get counted in surveillance data. And so that really allows us to monitor for chains of transmission that might be hidden, and try and go out and search them out so that we can control the outbreak more effectively. And in the last few minutes, I'm going to talk just a little bit about how we've been operationalizing genomic epidemiology for understanding SARS-CoV-2 and COVID-19. I think that, this is something we all care a lot about. It's, of course, the reason why we're talking on Zoom. And there's a lot of traditional epidemiology that's going on. You've probably heard a ton about contact tracing. But we are also doing genomic epidemiology to understand SARS-CoV-2. And in fact, it was the genomic data really right at the beginning that was one of the biggest red flags that we had very easy human-to-human transmission. And what we saw from the initial genomes from Wuhan and from Thailand was that there was very, very little genomic diversity there. A lot of the viruses were identical. And that is a major signal for human-to-human transmission, as opposed to repeat spillovers from an animal reservoir, say at the seafood market. And so this really is kind of what made us quite concerned, actually, back in January, that we had the potential of a massive outbreak because human-to-human transmission appeared to be so easy, which is not necessarily true for many viruses that spill over from animal reservoirs. We've also used genomic epidemiology to understand transmission in Seattle. So as some of you Seattle-based folks might have heard, there was this initial case that traveled to Wuhan and then came back to Everett and developed disease and was treated in like total isolation in one of the Ebola quarantine rooms and was treated with a robot. And we thought that that had stopped all onward transmission. But in fact, there was a teenager up in Snohomish County who participated in the Seattle flu study and was positive for COVID-19. And using the genomics, we were able to see that that genome was actually descended with a little bit more genetic diversity, a few more mutations from this initial sequence from the person who had returned from Wuhan. And so that is really what kind of clued us into the fact that probably SARS-CoV-2 had been circulating in Seattle during the month when we didn't know that it was there. And then I think something that is one of the coolest things to see is that once you bring technology like sequencing, which can work across pathogens, into a country and build capacity there, they can use it for a whole bunch of different pathogens. And so the genomic surveillance system that was being used for Ebola in the Congo is now also being used to sequence SARS-CoV-2. And what we see from the DRC strains is that they are most closely related to viruses sampled from France and Belgium, which is unsurprising given the sort of travel patterns between the countries. But it really kind of helps us to understand travel importation into the DRC as well as their epidemics on the ground. And I think one of the things that is really, really cool is that the DRC is now the most densely genomically sampled country for SARS-CoV-2 in Africa. And so I think that this capacity building really could revolutionize genomic surveillance and public health within some of these countries where that infrastructure might be lacking. So with that, I want to save some time for questions. So I will kind of swap over here, and I'm happy to talk about any of these viruses or path to science or anything like that.
And feel free to unmute yourself or put questions in the chat. We can take questions either way, but please go ahead and ask your questions.
And do you want me to stop sharing and just make it normal Zoom call?
Sure.
All right.
And we can see other people more easily.
That sounds good. Okay. Oh gosh. Stop sharing.
Great, thank you.
You bet.
I'll go as people are putting their video back on, but you mentioned that you need about 80% of the genome in order to add it to the tree. How hard is that to obtain? Yeah. Especially in places where you have to like pack all your science in a suitcase and truck it over somewhere.
Totally. No, that's a great question. I mean, I think you can absolutely build trees with less, and we routinely do. It's just that for a lot of these kind of more sophisticated analyses where you're asking a lot of the data, you get better inferences when you actually have more information in them. I'd say that You know, sometimes bringing and oftentimes bringing the lab to where the samples are results in better data quality. And the reason for that is the more that the samples are sort of brought up to a higher temperature and then brought back down and freeze thawed, as we call it, more they tend to degrade. So there's all sorts of stuff in our biological samples that can degrade pathogens, and they do that even in a test tube, as long as that is at a temperature where that can happen. And so generally, I think this is why so much of the focus has been on developing genomic surveillance capacity within country, so that you can sequence, right there where the samples are collected. And that's also why there's this push for mobile sequencing or field sequencing, is that it's a way to train people, and it's also a way to use the samples in the country. And that's important just from like a sample degradation thing, but also from like a preserving the autonomy of the country and not expatriating samples. It's sort of like an ethical thing too.
Great, we have a couple in the chat. How good is the coverage of sampling across the world? Are there areas that you are oversampling or undersampling?
So I'm assuming, Danielle, that is in relation to SARS-CoV-2. Yes, there is a huge amount of variation in the sampling intensity. There are places like Iceland, and they've sampled a huge fraction of their genomes, or of their positive samples. There are other countries, like I'm trying to think. I don't know that we have any sequences that were generated in Iran. I think we know a little bit about the sequence diversity from Iran just based off of people who traveled to Iran and came back, and then we were able to sequence their genomes. But yes, I mean, there's huge heterogeneity in which countries tend to be well sampled and which ones aren't. And that absolutely impacts our analyses and our ability to understand these sort of geographic questions about what countries tend to be directly transmitting to each other. I think that answers your question, but if it doesn't fully, please feel free to throw in something else.
There's also one, has the source of the SARS-CoV-2 virus that was found in the flu study person been traced back to origin? Like, do we know from genomic epidemiology likely where how that came to be?
Right. So I think there's like basically there's kind of possible, there's like two possible narratives. So the first one is that the individual who went to Wuhan and came back to Everett is the source of that entire Seattle clade. And we know about it now, and we've been monitoring it now, but there was that entire month-long period in February when we didn't know about it. And that, that seems relatively likely because the constraints on testing were quite strict during February, where you couldn't get a diagnostic test to see if someone had COVID-19 unless they had a travel history to Wuhan or they had contact with a confirmed positive case. And so you can imagine that breaks down where all it takes, so the person who was hospitalized in Everett, we know that they're a confirmed case. If they pass it along to someone else, or someone got infected by that person but was asymptomatic or only mildly symptomatic and didn't get tested, but they still transmitted it on to a third person, that third person now doesn't have a travel history to Wuhan and also doesn't have contact with a confirmed case of SARS-CoV-2. And so what that means is that with the sort of really tight specificity of that definition, it kind of ruled out our ability to go looking for the virus. And it wasn't, probably given the definition that we had surrounding testing at that point, we probably wouldn't have seen that until CDC had loosened up the testing if it hadn't been for the Seattle flu study. There's always the possibility that when the person came back from Wuhan and came back to Seattle, that those viruses were still circulating in Wuhan. And so it is possible that someone else from Seattle went to that same area, got infected with a similar virus, and came back to Seattle. It just seems very unlikely that that is the case, given sort of the other characteristics of that setting where, the teenager was also up in Snohomish County. And there's sort of like a lot of other factors that seem to show that those cases are likely related. But, there's other possible stories. I think that sort of like hard genomic evidence is just that the teenager has an almost identical genome sequence to the individual up in Everett with, I think it is 2 additional mutations, which sort of indicates the continued transmission that was undetected.
There are a couple more in chat now. One of them has to do with how much experience and coding you need in order to do your research. And I'm just kind of curious to broaden that about, if there are some students on this call, and if they were interested in this field, what would you say in terms of, good experience to have?
Definitely. So I will say that when I started my PhD, I did not know how to code. And that made the first year of PhD really hard, because not only was I trying to learn all the background science, but I was also trying to get my coding skills up to snuff. And so there is a lot of coding involved in this research, but I would highly encourage people that if you are interested in this work, coding is a skill like anything else that you can learn. And I think it's very easy, or at least I found it very frustrating at times. And I think it's easy to think that if you don't already know how to do it, that precludes you from a field of study. And I would just say absolutely not. In science, what really matters is how you think and how you ask questions. And the whole point of your training is to develop the skills that allow you to investigate those questions. So if you can think well about problems, then you can learn to code and it'll be fine.
Okay, we've got 3 more and about 5 minutes left. And
okay,
so
I'll try to do it faster.
no, I've got no pressure. We could just pick one, or, if we could try to do all 3, but what are some of the biggest insights you've had so far, and what questions are you asking at the moment in the Bedford Lab about this virus? The second one is, if there were different genome sequences found in one, I think, geographic area that was the origin of the disease, how do we know which one was the original sequence and which ones were mutations? And then the last, with both Zika and SARS-CoV-2, your data indicated the disease was transmitted for quite a while before people were aware of it symptomatically. Do you think that is a pattern that occurs with most viruses?
Right. Okay, so I'll answer Angeline's question first, I think. So this is actually one of the reasons why you want to have longer genome sequences, is that you have this sort of nesting pattern where you'll have new mutations accruing, but they're always accruing against sort of the backbone of the virus. So you can imagine that if I get infected with a virus and that there's a mutation that I transmit onto someone else, and they get a new mutation, they will have kind of the mutation that characterizes the virus that I passed along to them, as well as the new mutation that cropped up during their infection. And so you end up with this sort of nesting pattern where, there's, say, 40 viruses that all share a mutation kind of deeper in the tree, and then 20 viruses that share another mutation, and then 2 viruses that share another mutation. And so you're getting that is kind of how you can tell which one was probably one of the earliest sequences. I think that allows you to see sort of the pattern through time, but I do want to stress that there's never going to be this is the very first virus or anything like that. For most of the initial viruses that we sampled from Wuhan, they actually all have identical genome sequences, and that's because the virus doesn't mutate. We get on average about 1 mutation every 2 weeks. and people can actually transmit the virus more quickly than that. And so you could have pairs of infections that have identical genome sequences, and then from the genomic data, you have no idea kind of whom infected whom. And so there's sort of limits to the signal that you can draw from the genomic data. Margaret, with you're welcome. So with Zika and SARS-CoV-2, so I would say there's a little bit of a distinction here. In Wuhan, I think that the evidence is actually pretty good that it was not circulating in China. Really, like, I think the absolute earliest would be November, and it's more likely like mainly in December of 2019. And so that's actually pretty limited transmission prior to kind of notification of the syndrome, in comparison to something like Zika, where it was like a year. I think that, I think rather than this being like a pattern that occurs with most viruses, I think that this is really kind of connected with the fraction of cases that are asymptomatic. And I think it also has to do with kind of the capacity of surveillance systems in areas where diseases emerge. So when Zika was introduced to the Americas, Brazil conducted mainly what we call syndromic surveillance or things like that, where people had fever. And generally, if you have fever, at the time, you know, dengue and chikungunya were circulating. And so most people would say like, oh, you probably have dengue. And there wasn't necessarily a lot of sort of PCR-based diagnostic testing. And so there was no sort of like a double check as to whether it was dengue or it was chikungunya or not. And that's really hard. You know, like that's not Brazil's fault or anything like that. It's really hard to do surveillance that intensively and that accurately, and it's especially harder to do in lower resource settings. And so I think a lot of it is kind of more related to surveillance infrastructure, and that's a really key part of improving public health. All right. Jeannie, you wanted to say something at the end, right? I want to give you 1 minute or whatever.
Yeah, so, but, you know, if you want to briefly wrap up this question from Danielle about, you know, what's happening in the Bedford Lab, I want everybody to know, though, that you had just successfully defended your dissertation recently. So on top of thanking you for this talk, we should all be celebrating that you are now Dr. Allie Black. So
thank you so much. Thank you.
Yeah.
Super fun.
So what's happening like in the Bedford Lab? What's happening there now?
What is happening? Well, since the beginning,
you're probably absorbed, right, with all the stuff.
Yeah, literally, yeah. So we all work from home now. We've been doing that since the beginning of March, and I would say, kind of to varying degrees, we have all switched to COVID work. So I put another Zika paper that I was working on hold. Basically everybody in the lab has kind of put their other projects on hold to work on SARS-CoV-2. A lot of that is maintaining those next strain builds, which you can all, if you want to go to nextstrain.org, you can look at these trees yourselves. And we also have these narratives that describe what sorts of epidemiologic dynamics are apparent from the trees. So most of the folks in the lab are working really hard all the time to keep those builds updated and to write those new narratives to kind of explain the data. And I have been working a lot actually on sort of technologies for contact tracing and thinking about how we can also support contact tracing in addition to genomic epidemiology. But we're basically all working on SARS-CoV-2 at this point, and we'll return to other science once we're not in a pandemic.
All right, thank you so much. I just basically wanted to, in addition to, thanking you for this incredible talk, make sure that people know that we have other exciting topics coming up. Next week, Dr. Nina Salama, who's a faculty member at Fred Hutch and professor of human biology and public health sciences and basic sciences, will talk about can bacteria cause cancer? And for those of you who are teachers, we have a professional development coming up June 3rd, building a gender inclusive biology curriculum, which we can send you more information about if you're interested. And please do take a minute if you can to give us some feedback on our feedback form. There's a QR code and we also have it linked in the chat. But if you want to unmute yourself and say thank you and goodbye to your colleagues and to our speaker, now's the time.